Now we've learned about the parts of the nucleic acid. We've learned about we've learned about replication. Now we're going to talk about transcription. This is all about how we get from DNA all the way to a protein. It's the flow of genetic information and gene expression. And we're going to learn specifically now how to describe the process of transcription. Transcription, in a nutshell, is where you're going to use a template strand of DNA, and you're going to use RNA polymerase, as said earlier, to create a new strand of RNA. If DNA polymerase builds a new DNA, RNA polymerase is going to build a new RNA. Here are some really important terms that you're going to need to know and be able to use appropriately. The promoter, the terminator, sigma factor, RNA polymerase, template strands, and transcription factors. I'll give you a second to write those down. I had to point this is one of my favorite slides because I could not find a good picture of RNA polymerase that wasn't copyrighted, so I did mine with like finger paint. This is me finger painting biology. Here's the way this works. You've got a strand of DNA. You want to make RNA. This long strand of DNA is at the bottom. DNA and RNA polymerase don't really talk to each other well. You need something that will bring the polymerase to the DNA. In prokaryotes, that's called sigma factor. That's this bottom section right here. In eukaryotes, they're called transcription factors. They speed up the rate of transcription. You need something to attach the polymerase, the top part, the RNA polymerase, to the DNA. Attached to the polymerase region at the top, you got these two antennas. They're looking for signals that say, go faster, go slower. Don't worry, we'll get into that in the next lecture. You'll notice there's an alpha region here. This alpha region is going to interact with the DNA. That's the actual polymerase part. The beta region, the beta region is going to create the RNA. So over here in alpha, you've got a helicase action, and over here in beta, you're building RNA. Now, like I said, you have to get the DNA and the RNA polymerase to bind to each other. Sigma factor can do it, and so can a transcription factor. This is sort of like, you all have those friends that cannot find a date without you, right? Okay. Or you are that friend. I don't know. That's cool too. Whatever. Okay. Do you know who I'm talking about? You've all got that one person in your social circle who's like, hi, yeah, they don't have many social skills. They're really super timid people. Like, hey, can you introduce me to them? And you get to be that sigma factor. You are that transcription factor. You bring the RNA polymerase, which is your friend, over to the DNA, which is your other friend, and hopefully they connect to each other. Where they bind, they're going to bind before the gene at a region called the promoter. The promoter is a sequence of nucleotides that allows for the binding of sigma factor, allows for the binding of transcription factors to attach an RNA polymerase. So you've got these things bringing in You've got these transcription factors, or you've got sigma factor pulling in the RNA polymerase and attaching it to the DNA. Once they're attached, what do? Just like you would do when you hook your friends up, you get out of there. You're like, go, be fruitful. I'll just be here all by myself. And then you get to do the whole pity party thing. That RNA polymerase now loses sigma factor, or it loses the transcription factors and starts to run along the length of the DNA. As it's going, it's unzipping it. So it's opening up the DNA, allowing access to all those nucleotides. This means that polymerase can begin to create a transcript. A binds to U, A binds to U, C to G, G to C, C to G, C to G, G to C, and so on. That polymerase is producing this RNA based off of that strand of DNA. So that RNA polymerase is sliding down the template strand, producing a new molecule that's RNA. And it's based on DNA that's extremely similar. Instead of uracil, though, it's got thymine. And the final step is termination. Here's the RNA polymerase gets to the end of the gene. It finds a stop sequence and is released. It's a terminator sequence. The RNA polymerase will be released. The RNA itself decouples, and you have a free-floating piece of RNA. The DNA zips itself back up. So there are 3 stages here: initiation, elongation, termination. During initiation, the polymerase is able to bind with sigma factor or transcription factors at the promoter right before the gene. Elongation, that polymerase runs along the length of the DNA, producing a molecule of RNA. In termination, where the RNA polymerase and the RNA are released from the DNA. The DNA zips itself back up. Everybody moves on their way. Everybody's happy. With transcription, we've taken a piece of DNA and transcribed it to the language of nucleic acids and converted it to RNA. Not converted it, we've just created an RNA strand from a DNA template. We've made sort of an imperfect copy. We're using ribose instead of deoxyribose. We're using uracil instead of thymine. Now, this transcription is occurring in the cell's nucleus. So you have the DNA in the nucleus, the transcription is occurring in the nucleus, because the DNA doesn't leave the nucleus. RNA polymerase is going to bind to DNA, and that polymerase acts as both a polymerase and a helicase, so it's splitting it and copying it at the exact same time to create a corresponding RNA molecule. And then that RNA molecule is really a temporary copy of the gene that has the instructions from the DNA. That RNA will leave the nucleus and go to the ribosome, where protein synthesis will occur. Protein synthesis is called translation. Transcription is really essential to this. Now, we haven't really made mRNA yet. We hear a lot about mRNA. In our next mini lecture, we'll talk about mRNA and how it's formed. These content review questions are here to focus your studies. The next mini lecture, like I just mentioned, will be about RNA processing. So what happens between when RNA is produced in the nucleus and when it goes out to the ribosome, because we're going to do some things to it to make it be able to live in that cellular environment outside of the nucleus. We'll have to add a little bit of some markers onto it. We'll have to remove some pieces of it to make it nice and lean and perfect. So the next mini lecture will be on RNA processing. Coming up now.